Hello students, today we can see the topic mitosis, a type of cell division. Where this mitosis take place? Mitosis occurs in somatic cells. Okay, mitosis occurs in somatic cells. What are somatic cells? These are nothing but the body cells. All the body organs are made up of the cells called somatic cells. Okay, except the reproductive cell, all the other cells we can consider it as the somatic cell. So in the somatic cell, mitosis division takes place. Okay. Mitosis is also said to be as equational division. Why it is said to be as equational division? Because when the cell divides, it divides in such a way that the chromosome number is kept the same in daughter cell equal to that of the parent cell. So the daughter cell are quantitatively as well as qualitatively similar in genetic material to that of the parent cell. Okay, that is the number of chromosomes in the parent cell as well as the daughter cell or the progeny cell is the same. So mitosis is also called as equational division. Okay, the duration of mitosis will be varying from 30 minutes to 3 hours. Okay, it ranges from 30 minutes to 3 hours. In different organism, it will be different in time. Okay, in human beings, in our somatic cells, the duration of mitosis is about 1 hour. Okay. Now, when mitosis occurs, first nuclear division takes place, followed by that the cytoplasmic division occur. Okay. So first process is karyokinesis. Karyon means what? Nucleus. Kinesis means division. So first nuclear division occurs, followed by that cytokinesis occurs. Cyto means the cyton, the cell, cytoplasm. Kinesis is division. So division of cytoplasm occur. Okay. So first in mitosis or in any such divisions, any such cell division, first the nuclear division, that is karyokinesis occurs. Okay. Karyokinesis occurs in 4 stages. Okay. What are those 4 stages? First is prophase, then metaphase, anaphase, and then telophase. Prophase is the first stage of karyokinesis of mitosis. In the S phase of interphase, we have seen that synthesis of DNA molecule occurs, isn't it? So the DNA molecule will be doubling. And these DNA molecules are not distinct, but they are intertwined with each other. And where these DNA molecules are present, they are present within the chromatin material. What is a chromatin material? It is a thin, long, thread-like structure which is seen inside the nucleus of a non-dividing cell. So during prophase, what happens? Condensation of this chromatin material occurs. What is that condensation of chromatin? The chromatin, which is long, thin, thread-like structure, will get shortened by coiling and it becomes a rod-like structure called a chromosome. Okay, so during prophase, the condensation of chromatin material begins so that it becomes a chromosome. Okay, so if you see in this picture, you can see the chromosomes. It is a rod-like structure. You can see the rod-like structure of this chromosome, which is having 2 chromatid. This is one chromatid, this is another chromatid, and they are attached in the center by means of a centromere. Okay, so that is the primary constriction, and that constriction is called the centromere, and the 2 chromatids which is longitudinally split. Okay, you can see the longitudinal chromatids. These 2 chromatids will be attached at the region of the centromere. Okay. Now, in the S phase, we have also seen that in the animal cells, the centrosome will be splitting into 2 centrioles and it will be moving to the opposite poles, isn't it? So, here you can see this is one centriole and this is another centriole. So, these 2 centrioles are moving towards the opposite poles. Okay. Then, from the centrioles arises the aster. So, here you can see the aster. Okay, the star-like structures which you are able to see here is the asters. They are formed of the microtubules. So, what is this microtubule? In the chapter Cell, the Unit of Life, you have studied that in the centrioles, the microtubules are formed, isn't it? The microtubules are filamentous proteins. Okay, they are protein filaments or protein fibers. Tubular proteins are said to be as the microtubules. Okay, so these microtubules will form the asters. Okay, and from this asters arises the spindle fiber. So here you can see this long, these are the spindle fibers. So these spindle fibers arises from this asters. So the two asters together with the spindle fibers form the mitotic apparatus. Okay. Now, in addition to these events that take place during prophase, the nuclear envelope disappears. Okay, that is another event. So here you can see this is the nucleus, isn't it? This is the nucleus and it is surrounded by the nuclear envelope. But here and there you can see the nuclear envelope is not there. Here it is missing. Here it is missing. That is this brown colored is the nuclear envelope. Okay, so you can see it is disintegrating, isn't it? And slowly it disappears. By the end of prophase, it will be disappearing. The nuclear envelope disappears. And here a light round small structure, light colored structure is there. That is what nucleolus. So the nucleolus also disappear. Okay, so by the end of prophase, we can see nuclear envelope and nucleolus disappear. Then some of the cell organelles like Golgi complex, endoplasmic reticulum also disappear. Okay, so these are the events that take place during prophase. Metaphase is the second stage of karyokinesis of mitosis. Metaphase begins with the complete disappearance of the nuclear envelope. The condensation of chromosome is also completed in metaphase stage. As a result, when we observe the chromosome under the microscope, it can be clearly seen. Okay, because maximum condensation of chromosome occurs during metaphase stage. Okay, so in this picture, if you see the chromosome, you can see the maximum condensation has occurred and the chromatids are clearly visible, isn't it? The chromatids here on chromatid, this chromatid you can see it clearly visible and it is attached in the center by means of this centromere, that is the primary construction. Okay, now on the surface of the centromere, disc shaped structures are there. They are called as kinetochores. Okay, on the surface of the centromere, disc shaped structures called kinetochores are present and all the chromosomes will be getting aligned on the metaphase plate. What is metaphase plate? The plane of alignment of chromosomes during metaphase at the equator is called metaphase plate. The plane of alignment of chromosome at the equator during metaphase is called metaphase plate. Okay, so you can see alignment means arrangement in a line. Okay, so here you can see all the chromosomes are arranged in a line at the equator. Equator means the center of the cell. Okay, equator here means the center of the cell. All the chromosomes are arranged and this is the metaphase plate. So all the chromosomes will be arranged at the metaphase plate. Then you can see the spindle fibers, isn't it, which are arising from the centrioles. They will be seen attached with the kinetochore. Okay, so the disc shaped structure is there in the centromere, so you can see them attaching to the kinetochore. On either side it will be getting attached. Okay, the spindle fibers get attached to the kinetochore of the centromere. Okay, so these changes takes place during the metaphase stage. Anaphase is the third stage of karyokinesis of mitosis, and during anaphase, the centromere will split and the chromatids separate and the chromatids will be moving to the opposite poles. So in the previous one, we have seen that the chromosomes will be like this, isn't it? The chromosomes will be having the chromatids. Two chromatids are there which are attached here at the center by the centromere. Okay, Now, what happens during this anaphase state? This centromere will be splitting into two longitudinally. So, like this, it will be splitting. As a result, the two chromatids, this is one chromatid, this is another. The two chromatids will be separated. Okay, longitudinally, they will be separating. And now, these daughter chromatids, we can also say it as the daughter chromosomes. Okay, after splitting. Yes, now after splitting, they will be moving towards the opposite pole. Okay, how it moves to the opposite pole? You can see the spindle fibers attached to the kinetochore of the centromere. On either side you can see that, isn't it? Now this spindle fiber will be contracting. So when it contracts, it will be pulling this chromatid towards the poles. Okay, so it will pull the chromatid towards the pole. Thus the chromatids will be moving towards the pole. You can see the centromere will be facing towards the poles and the arms. This is the arm of the chromatid. It will be trailing behind. Okay, trailing, it will be moving behind. Okay, so like this, the chromatids will be pulled towards the opposite pole, and now the daughter chromosomes will be reaching the poles. Okay, so the number of chromosome is not getting changed here. The same number of chromosome is remaining on both the sides, isn't it? The same. Okay, here you see 1 2 3 4, and this side also 4 are there, and the parental in the parental cell also we have shown that there are 4 chromosomes, isn't it? In the previous slide we have seen that there are 4 chromosomes. Now in the daughter cells also they will be getting 4 chromosomes only, because each chromosome have split into 2 based on the doubling of DNA which has taken place. Okay, now it is splitting into 2. As a result, each pole of the cell is receiving the same number of chromosome. Okay, so the number of chromosome is not at all getting reduced. The same number of chromosome is retained in the daughter cell. That's why this mitosis is said to be a sequential division. Okay. Telophase is the final stage of karyokinesis. During telophase, the chromosomes reach their respective poles and then they decondense. What is decondense? The chromosomes now become chromatin material. Okay, so here in this nucleus you can see in each nucleus 4 chromosomes are there. So clearly it is seen as chromosomes, isn't it? But now it becomes decondensed to form chromatin material by losing their identity as discrete elements. Discrete elements means a distinct or a separate element. Element here means what? The chromosome. They were seen as separate chromosomes, isn't it? But when they decondense, they become again thin, long, thread-like structure, so that we cannot differentiate it into a chromosome. They all will be seen as entangled mass of chromatin material. Okay, and what are the other changes that take place? Nuclear envelope will reappear, will be reformed. Okay, and nucleolus also will be reappearing, and the organelles which were lost like the Golgi complex, endoplasmic reticulum, etc. also will be reappearing. Okay, so these are the events that occurs during telophase stage. As a result of karyokinesis, two daughter nuclei are formed, followed by karyokinesis, cytokinesis occur. What is cytokinesis? It is the division of cytoplasm. As a result, the single cell will be divided into two daughter cells. Now, how the cytokinesis occurs in animal cell and plant cell, we can see. In animal cell, the cell is surrounded by a cell membrane, isn't it? So, what happens? A furrow appears. Here you can see a furrow appears in the plasma membrane. What is meant by furrow? It is nothing but a depression occurs. So you know that the animal cell is surrounded by a plasma membrane. So first a furrow appears like this. Okay, now this furrow will be deepening. So it deepens like this. Okay, and at last it divides the single cell into two daughter cells. This is how the cytokinesis occurs in animal cell. Okay, but if you take a plant cell, in plant cell the cell is surrounded by a cell wall. isn't it? Plant cell is surrounded by a cell wall. So here the cell wall, as it is very rigid in nature, a furrow cannot be formed. So a cell plate will be formed in the center of the cell. What is that cell plate? It is formed by the accumulation of vesicles formed from the dictyosomes, which contains pectin, cellulose, hemicellulose, etc. So they all will get deposited in the center of the cell. Then it will be deepening towards the peripheral region, and thus it separates or divides the single cell into two daughter cells. Okay, so this cell plate will later develop into the middle lamella. What is middle lamella? It is a layer which is present between the cell wall of two adjacent cells. Okay, the layer which is present between the cell wall of two adjacent cell is called the middle layer, middle lamella. So this middle lamella is formed from the cell plate. Okay, thus the plant cell will be divided into two daughter cells. Then some of the organelles like mitochondria and plastids, which are present inside the cell, will also be dividing equally, and they get distributed into the two daughter cells. Okay. Now, in some cells, after karyokinesis, it will not be followed by cytokinesis. As a result, what happens? A multinucleate condition will be existing. You might have studied in lower organism like fungi, aseptate condition is there, that is syncytium will be formed, isn't it? So what is that syncytium? A mass of cytoplasm with many nuclei. So such conditions will be formed. If you take the coconut, you all are familiar with the coconut. It contains the liquid material as well as the solid kernel. White kernel will be there, and a fluid also will be there in the coconut, isn't it? So that fluid is actually the liquid endosperm, and the white colored kernel is the cellular endosperm. That means in the beginning, in the tender coconut, if you take, everything will be fluidy in nature, isn't it? Actually that fluid is nothing but the endosperm only. And here the endosperm is only formed of nucleus, that is nuclear endosperm. Okay, what is nuclear endosperm? The nucleus is not surrounded by a cell wall. So when cell wall is not there, we can say it as a nuclear endosperm. Okay, later only slowly cell wall will be laid around the nucleus, and as a result the white kernel will be formed. The white part of the coconut which we eat will be formed. That is actually the cellular endosperm, that is the nucleus is now surrounded by the cell wall. Okay, as a result cellular endosperm is formed, and the liquid endosperm which is present is the nuclear endosperm which is not surrounded by cell wall. Okay, so when karyokinesis is not followed by cytokinesis, then multinucleate condition will be existing. Okay, so that's all about the mitotic cell division. Next, we can see the significance of mitosis. So, as a result of mitosis, a single cell divides into two daughter cells. The two daughter cells will be diploid in nature, that is, it will be having two set of chromosomes just similar to that of the parental cell. Okay, and the cells which are produced are genetically identical to the parent because there is no genetic recombination taking place during mitosis. Okay, so from a single parent cell, two daughter cells will be formed which is diploid in nature. Okay, so the first significance of mitosis, we can say they produce genetically identical daughter cells. Then another significance is the growth of the body occurs through mitosis. Okay, the single celled organism or the multicellular organism, all organisms develop from a single cell, isn't it? Even we develop from a single celled zygote. So the single cell will undergo mitotic division to form 2 cell, then 4 cell, 8 cell, 16 cell, like that it goes on dividing, and a mass of cells will be formed which later gets differentiated into different tissues and organs, and thus an organism is formed, isn't it? So for the growth of the body of an organism, mitosis is very essential. Then for cell repair and replacement also, mitosis is essential. Okay, so what is that cell repair and replacement? See, suppose if a cell get damaged, if an injury occurs in an organism, then that injured cell or that dead cell has to be replaced by a new cell, isn't it? For that, which division occurs? Mitosis occurs in the neighboring cells, and thus daughter cells will be formed and they will be replacing the dead cell or the damaged cell. Okay. If you take our epidermal layer, that is our skin, always a layer of cells will be damaged and slowly it gets eliminated, isn't it? But new and new cells are formed and it is replacing the damaged cell, isn't it? It is by the process of mitosis. Same way in our digestive system, in our mouth, when we take hard food and chew the food, the layers lining the gut, the mouth will be damaged, but again it will be replaced, isn't it? Even our blood cells are also, their lifespan is very short, so they will get damaged soon, and again new and new blood cells are being produced by the process of mitosis. In plants also you can see the apical meristematic tissue is there, lateral meristem, that is the cambium, is there. So they also are dividing and producing new cells as a result of mitosis. Okay. Then another significance is maintenance of nucleocytoplasmic ratio. A nucleocytoplasmic ratio should be maintained in each and every cell. It should be restored. So that can happen due to mitosis. When a cell is dividing, then the daughter cell will be having the correct nucleocytoplasmic ratio. Okay, so for maintaining that nucleocytoplasmic ratio also, mitosis is essential. The normal nucleocytoplasmic ratio is 4:1. Okay, 4 is the ratio of nucleus and 1 for cytoplasm. That is related to their volume. This nucleocytoplasmic ratio is given. Okay, actually these are essential for the formation of mitotic spindle or the formation of metaphasic plate. So for all that it is essential. Actually it is an event of mitosis itself, but that is essential for maintaining the mitosis itself is essential for maintaining the nucleocytoplasmic ratio. Okay, so that is about the significance of mitosis.